Hello everyone, this is Dr. P. Mary Anupama, Head of the Department of Biochemistry, St. Joseph's College for Women, here with the topic Mechanism of Nerve Impulse Transmission. The contents of today's lecture include basic introduction, followed by learning about resting membrane potential, then what is action potential, its initiation, the avalanche effect, followed by repolarization and restoration after threshold, then action potential curve, followed by propagation of nerve impulse and saltatory conduction, and then the events at the motor end plate. We are aware that the nerve impulse transmission, it's a complex physiological process that enables communication within the nervous system and also between the nervous system and the effector organs. It involves both the electrical and chemical events that ensure the rapid and precise response to stimulus. This process, it can be broadly divided into three major stages. The first one is the establishment of resting potential and initiation of action potential, propagation of impulse through saltatory conduction, and then transmission at the motor end plate. Understanding these steps, it will provide insight into how the body coordinates movement, sensation, and even high cognitive functions. Resting membrane potential. At rest, the neuron, it maintains a membrane potential of minus 70 millivolts. When you check the membrane potential across the membrane between the outer and the inner, it is minus 70 millivolts. This negative internal environment, it is because of the unequal distribution of ions, which include the sodium ions, potassium ions, as well as chloride ions, and even proteins. The sodium potassium pumps, like the Na plus K plus ATPase, they play a very crucial role. These pumps, they actively transport 3 sodium ions out of the cell and allow 2 potassium ions into the cell at the expense of ATP. Additionally, the membrane is more permeable to potassium ions than to sodium ions due to the presence of potassium leak channels, allowing these ions to move out of the cell more readily. So this creates a negative environment inside the neuron. Thus, the resting potential is established by a combination of active transport and passive diffusion mechanisms. It represents a polarized state of the neuron, preparing it for the generation of an active potential when stimulated. Action potential initiation and avalanche effect. When a neuron is stimulated, it undergoes a rapid change in the membrane potential, and this we call it as the action potential. This begins when the stimulus, it depolarizes the membrane to a critical level, which we call it as the threshold potential. It is approximately minus 55 millivolts. At this point, the voltage gated sodium channels, they open, allowing rapid influx or rapid rush of sodium ions into the neuron. This causes further depolarization. The depolarization, it turns open more of sodium channels, and this positive feedback mechanism, it is referred to as the avalanche effect, where the entry of sodium ions, it is accelerated, and the opening of additional channels will be taking place, leading to a sudden and steep rise in the membrane potential. So as a result, what we see is the membrane potential, it quickly shifts from a negative to a positive, and this is known as depolarization. Okay. The action potential, it follows the all or none principle, measuring that once the threshold is reached, the response is maximal and it is independent of stimulus strength. In this image, we can see the depolarization. So here, when stimulus is given to a neuron, the sodium channels, they open up, causing a rapid influx of sodium ions. So as the sodium ions open, the depolarization initiates and it spreads across the membrane. This we call it as the avalanche effect. So this happens to some extent only. After some time, as the membrane reaches a saturation state, which we call it as the action potential, it is stopped, and then it starts coming to the resting state, that is back to the resting potential, which we call it as repolarization. So what is repolarization and how it is restored after threshold? After the peak of the action potential is reached, we have seen that the neuron must turn back to the resting state. This involves some coordinated steps. First is the voltage gated sodium channels. They become inactivated, which stops the sodium ion influx. Simultaneously, voltage gated potassium channels, they open, and this allows the potassium ions to diffuse out of the cell. So the outward movement of potassium, that alone restores the negative charge inside the neuron, and this phase we call it as repolarization. So here the membrane potential, it becomes more negative than the resting potential, resulting in what is known as hyperpolarization phase, where there is excess potassium ions loss. So what we see is eventually all the channels, the potassium channels, they close, and the membrane, it tries to return to its resting potential. So here we have the sodium potassium pumps which get re-established, and then ionic gradient happens. So this restoration, it ensures that the neuron is again ready for the subsequent impulses. Additionally, the refractory period, it is comprising of the absolute and relative phases. We say absolute and relative phases, and the immediate reinitiation of another action potential starts. So this ensures that the impulses, they travel only in one direction along the axon, and it maintains the integrity of the signal transmission in one direction. That's why we call it as one-way impulse conduction. One-way impulse conduction. So repolarization explained through diagram. The membrane potential, it has to return to the resting state again. So we have lots of sodium inside and less of potassium outside. So what we see is the sodium potassium pumps, they open, and for every 3 sodium that move out, 2 potassium ions, they enter inside, and then the cell finally comes to the resting state. So once it has come to the resting state, it is again ready for the next set of impulse. So here this is the potassium channel which allows inward movement of potassium ions. As the potassium ions move inside, okay, we see that it has come back to the resting potential state. Action potential curve. An action potential curve, it represents the rapid changes in the membrane potential during a nerve impulse transmission. At resting stage, a neuron is said to have the resting potential which is around minus 70 millivolts. Upon stimulus, the voltage gated sodium channels, they open up, causing depolarization. And this depolarization, we call it as the avalanche effect, because there is a lot of rush of sodium ions into the membrane, which leads to a shift in the potential, and it reaches action potential. The maximum, we call it as the action potential, which is around 55 millivolts. Once it has reached this state, again it has to come back to the resting potential state. So this phenomena, we call it as repolarization. This is repolarization, while this is repolarization, depolarization and repolarization. So here what happens is, once it has reached here, now again the sodium potassium channels, they open up, and there is active transport of sodium ions out of the cell, and two potassium ions into the cell will be taking place. So this restores back the resting potential to minus 70 millivolts. So the representation of changes that are taking place in a graphical method, we call it as the action potential curve. Propagation of nerve impulse and saltatory conduction. Once the nerve impulse is initiated, the action potential propagation across the axon will be taking place. In unmyelinated fibers, usually this occurs through continuous conduction, whereas in myelinated ones, it occurs by saltatory conduction. And this happens because of some sequence of events, that is depolarization and repolarization, that will be continuously taking place across the length of the axon. So the axon in myelinated fibers, it is covered by myelin sheet, which is formed by the Schwann cells, and this we find in the peripheral nervous system. Or when you are talking about central nervous system, it is the oligodendrites that form the myelin sheet. This sheet, it acts as an insulator, electrical insulator, that prevents leakage of the ions, and it increases the resistance of this membrane. So the interruptions in the myelin sheet, which we call it as the nodes of the Ranvier. Here we have the nodes of the Ranvier. So at these nodes of the Ranvier, it is usually rich in voltage gated sodium and potassium channels. During saltatory conduction, what we see is that the action potential, it effectively jumps from one node to the other node, and then it travels continuously along the length of the action. When the action potential is generated, say for example at the axon hillock, when the action potential is generated, there is sodium ions influx and potassium ions will be going out. Okay, so this is a depolarization event which has taken place. Then as it moves, again here repolarization is initiated. Once a threshold of minus 55 millivolts is reached here, then again it comes back to the normal state. So what you see is, once this has initiated, it spreads along the length of the membrane. So here we are seeing the saltatory conduction, how it is jumping and all. It goes very fast. So once the depolarization is initiated here, the signal, it jumps to the next one. So once it jumps to the next one, here repolarization is initiated. So what you see is, as the saltatory conduction is taking place, it is a continuous stimulus which takes place. So initial depolarization followed by repolarization. Again depolarization followed by repolarization. So a continuous cycle of depolarization and repolarization events will be taking place because of the continuous stimulus that these cells receive. So this speeds up the pace at which the signal is jumping, and also it reduces the energy use because only these channels will require the energy, while the other channels which might be present here, they will not be requiring because they don't open at all. So this is how saltatory conduction, it makes sure that the nerve impulse conduction is done very fast and it is also energy efficient. So at the depolarization, action potential will be reaching here, followed by again resting potential. So that's how the depolarization and repolarization events make sure that continuous conduction of nerve impulse is taking place by the neurons. Now let us once again study about the events at the motor end plate clearly. Events at the motor end plate. This we also call it as the neuromuscular junction. So here the nerve impulse is transmitted from the neuron and to the muscle fiber. As the action potential it reaches this axon terminal, here we have what are known as the voltage gated calcium channels. They will be present across the axon terminal. So here the depolarization process opens these calcium channels. As the calcium channels open up, there will be calcium influx which will be taking place. This calcium influx, it will draw the synaptic vesicles towards the presynaptic membrane, and once the vesicles fuse with the presynaptic membrane, they release acetylcholine by exocytosis. So once the acetylcholine is released into the synaptic cleft, then it reaches the postsynaptic membrane, which is the muscle membrane. So here what it does is the acetylcholine which is present, it will bind to the nicotinic acetylcholine receptors which are present at the motor end plate, that is the membrane of the cell membrane, the muscle cell membrane. Now the membrane potential gets transferred to the muscle membrane. So the receptor, once it binds to this acetylcholine, what happens is they start opening up. They start opening up. And here the channels that open up, they are the ligand gated sodium channels. Once they open up, there is rapid influx of sodium ions that is taking place. Okay, and this initiates the depolarization at the muscle fiber. So we know that the muscle fiber, it has to transmit the action potential into it. And the muscle membrane, it has some extended tubules which we call them as the T tubules. So the action potential extends till here. As it extends, what you see is here the calcium which is present in the endoplasmic reticulum, that is the sarcoplasmic reticulum, that calcium ions will be released. And these calcium ions, they are responsible for the contraction of the muscles. So that is how once the action potential reaches the muscle membrane, it is transmitted via the sodium ions through the microtubules to the sarcoplasmic reticulum. The sarcoplasmic reticulum releases the calcium ions, and the calcium ions, they will be responsible for the muscle contraction. Now we have to study about acetylcholine molecules detachment. So the acetylcholine which is present here, it has to be removed again. The receptors are to be restored. So what happens is these channels, they close up, and we have what is known as the enzyme acetylcholine esterase. So this acetylcholine esterase, it clears all the acetylcholine which is present here. It hydrolyzes all the acetylcholine at the synaptic cleft, and this process alone ends the procedure. And the choline, it is taken back into the axon terminal for reuse. Now the motor end plate, it is ready again for the next impulse. So once again the cycle continues. The cycle will continue. So let us summarize here. Signal journey, we have 8 steps, right? So the signal, it is initiated in the axon, okay, the axon hillock. Then it reaches the axon terminal. Then calcium influx will be taking place at the axon terminal knob. Then acetylcholine is released. Then you see that the acetylcholine binds to the receptors. Once it binds to the receptors, we see that it results in initiation of action potential at the muscle membrane. So once the muscle membrane action potential is initiated, then it causes the calcium ions release at the muscles, then muscle contraction will be taking place. But parallelly what we see is the acetylcholine, it again detaches from here, from the receptors, and then it is cleared by acetylcholine esterase. Let us summarize what we have studied. The mechanism of nerve impulse transmission. It begins with the establishment of resting potential, membrane potential across the neuron. Then when it is stimulated, the neuron, when it is stimulated, it triggers depolarization, and you see avalanche effect followed by action potential establishment. Then again repolarization is initiated, which restores the resting state. We see that there is a lot of initially sodium ions efflux and potassium channels inactivation will be taking place. Then the impulse, it propagates rapidly across the axon through saltatory conduction in myelinated neurons. We have seen that. We have seen the events at the motor end plate and how the neurotransmitters, they enable signal transmission to muscle cells. Then we see that all this coordination, it ensures rapid, precise communication for sensory, motor, as well as cognitive function. Recommended books for further study include Guyton and Hall Textbook of Medical Physiology, Essentials of Medical Physiology by K. Sembulingam and Prema Sembulingam, or Human Anatomy and Physiology by Elaine and Merab et al. Or you can study Review of Medical Physiology by Barrett, Susan, or this is the Indian other one, Textbook of Medical Biochemistry and Physiology by M.N. Chatterjee and Rana Shinde. Thank you.